you very much, Summer, for the kind introduction, and thank you very much to the organising committee for this EpIC series of seminars. Just to kind of, as we say, Summer's introduced my talk already, but it's kind of originally being from London, Mind the Gap is kind of something that those may not be familiar with, is what's currently or consistently mentioned on the Tube network in London. Just as an outline kind of for my talk today, I'm just going to really discuss briefly some ion channel control of cerebral blood flow, especially in arterial smooth muscle cells. Then I'm going to move on to the main part of the talk, which will be the kind of the nanodomain regulation of RyR receptors and how these are functionally coupled to BK channels. And as Osama just mentioned, we've moved recently into dementia and really going to look at the consequences of impaired coupling on brain health. So just in case people need to leave, I suppose just wanted to start with some take-home messages as well, is that constant brain blood flow is maintained by a process called autoregulation. And this is where the arteries and arterioles modulate their diameter to maintain that constant volume of blood reaching the brain. The BK channel maintains essentially a larger vessel diameter and therefore sufficient blood flow to the brain. And as we'll get into, there's a real complex structural and functional regulation of this nanodomain and actually how this influences arterial tone. We'll touch at the end on some recent work in hypertension where we know this is shown to be damaged, this nanodomain. And then a recent work, or what we're trying to do now, is actually restore healthy blood flow and see whether that correct that underlying ion channel dysfunction and see if this slows disease. But I thought I'd just start with the brain. Obviously, not everyone here is a vascular physiologist, so we're kind of trying to ease in. And so you may be surprised to see, but actually there's over 400 miles of blood vessels within the brain. It's organized into kind of a hierarchical structure. Hill surface is where vessels run along the surface of the brain before diving deep into the actual neuronal tissue itself. These are known as parenchymal or penetrating arterioles, before giving rise to this very dense capillary network, which is essentially the majority of the circulation within the brain. And it's always interesting to see that actually there's more vascular cells in the brain than there are neuronal cells. And this is mainly due to almost a one-to-one relationship between a capillary and a neuron. But what actually drives blood flow in the brain and how is this modulated? Well, just starting very simply, blood flow is a simple process of the difference in pressure divided by the resistance that is to that flow. And where is resistance actually maintained or sort of generated? There's a real drive within the vascular community, especially in the cerebral circulation, about different areas of cerebrovascular resistance. And this is an overview from Idacola Lab a few years ago now, where a review where they just showed essentially the different aspects of that. So here they have the percentage of resistance as shown by, say, the large cerebral arterioles, given around 40%. This includes the middle cerebral artery and the kind of larger vessels that are on the blood vessel brain surface. Another 20% essentially is the pial arteriole, so the branches off of the main vessels on the brain, giving a rise to about essentially 60% of the resistance is actually outside of the neuronal tissue, with the rest actually comprised of those penetrating arterioles that I mentioned and the capillary network, with many other contractile cells down at the capillary level, including pericytes. Well, what I'm going to concentrate on today is the pial circulation. This is where I've been doing most of my work recently, or pretty much most of my work since my original postdoc. And we're just going to go through that cerebral autoregulation. But before we get there, I just wanted to kind of bring everyone back to Purcell's law. So I spoke originally about resistance and how that modulates blood flow. But really, Purcell's law kind of puts that in a bit more detail, where we can actually bring in factors that actually mean stuff, and not just resistance itself. And so you might remember this from your physiology courses. Hopefully many of us know what this is, but essentially it's all due to this equation where we can get differences in radius, pressure, fluid viscosity, and length will all influence blood flow. Well, what I'm really interested in is obviously the radius, as Osama mentioned before, and that's mainly due to, obviously, it's a strong power to the power of 4, some influence, therefore, on blood flow, but also there's a moment-to-moment changes that maintain correct blood flow over a range of pressures and moment-to-moment regulation. And this is the process of autoregulation. This is where blood vessels can change their internal diameter, as shown at the top, in response to intraluminal pressure or the blood pressure onto the vessel. What we can see as in this autoregulatory window, you can see as the blood pressure gets slightly higher, you can see that the blood vessel will slightly constrict, and therefore to maintain the correct amount of flow through that system with an increase in pressure. Whereas if the pressure drops, the blood vessel would dilate in order to maintain that correct amount of flow. And so this is the process that I'm really interested in. How do blood vessels sense that pressure? How do they respond and know exactly what arterial diameter to maintain? And obviously, being a moment-to-moment process, is really influenced by ion channel activity. And I hope this is not too rudimental for everyone, but I wanted to make sure everyone has a level playing field to start with. And this is how smooth muscle contractility is controlled by ion channels. So in that autoregulatory window, we might have a semi-contracted blood vessel, as shown here. With a semi-contraction, it's obviously a muscle cell. The contractile state is driven by the amount of calcium within the cell. The main source for calcium for contraction in smooth muscle cells is the calcium influx through voltage-gated calcium channels, as shown here in the blue channel. And this is really influenced by membrane depolarization and hyperpolarization between the two. But what I thought was really quite important for this audience as well is actually the membrane potential that we'll get to in a sec. But we're mainly looking at around minus 45 millivolts in a semi-contractile vessel with a calcium concentration around 220 nanomolar. If we was to depolarize the cell and drive a full constriction, we're only actually going to depolarize the cell by about 10 millivolts and increase the calcium concentration by another 100, 150 nanomolar in calcium to get a full constriction. Now, this change in diameter can have a massive change in blood flow downstream, and so there's a real tight regulation between the two ion channels. And just going the opposite way as well, if we was to hyperpolarize the cell by, again, maybe 10 millivolts, really drive down the intracellular calcium concentration and get a fully dilated blood vessel, which will maximally change or increase the amount of blood flow to the downstream brain in this case. And so there's a real tight regulation between depolarization and hyperpolarization. Many different ion channels are involved in smooth muscle contractility, and then really playing this sort of delicate balance to maintain the correct diameter in response to different inputs. Now, as you might have got from the title, I'm really interested in the large conductance calcium-added potassium channel, or the BK channel. This is a huge conductance channel, as everyone may know, around 240 picosiemens, and it's both calcium and voltage dependent. It's activated by calcium release events known as calcium sparks, which I'll get into in a sec, but this is calcium release from the sarcoplasmic reticulum in close proximity to the BK channel in order to generate sufficient calcium concentration to activate the channel. And really, as I mentioned before, this acts as a negative feedback system to hyperpolarize the plasma membrane, reduce the amount of calcium influx through voltage-gated calcium channels, and then cause a larger diameter of the blood vessel. And this is just some biophysics of the BK channel in mouse cerebral arteries. This is mainly what I use. So mouse pure cerebral artery, smooth muscle cells, and these are freshly isolated cells where I've just characterized the BK channel just to show essentially that's what we're recording. As you can see here, this is a inside-out patch where we've exposed the cytosolic region to 10 micromolar calcium, showing a huge amount of open probability and openings of this channel as the time goes on. And then you can see we've worked out the conductance is around that 240 picosiemen range. But when we look at what's really interesting is that calcium sensitivity. What you can see here in the log molar calcium is that at the sort of 1 micromolar, sort of 3 micromolar concentration, there's really little activity at all, but it's really this kind of switch on effect as soon as you hit 10 micromolar calcium. As you can see, there's this big jump in open probability, which really kind of drives this on, is either on or off channel that we kind of think about. And so really, we've got to get to around 10 micromolar calcium, if not higher, in order to activate the channel within this physiological membrane potential. And just to show that also this channel is voltage sensitive as shown here. Now, how do you generate that calcium concentration so high within a vascular smooth muscle cell when we're looking at a global calcium concentration of, say, 200 to 300 nanomolar? Well, you have to activate calcium sparks. Now, calcium sparks, as shown in this video here, are these localized calcium release events that look very much like fireworks, very apparent, seeing it's almost bonfire night here in the UK tomorrow. And these are these flashes that are shown here in these pressurized cerebral arteries that have been loaded with a calcium indicator. And as you can see in real time, every cell is pretty much flashing away, multiple sites per cell, reaching this high concentration, very transient releasing calcium, which is close proximity to the plasma membrane, in order to activate that BK channel. But how do we get that kind of concentration so high? Well, we have to essentially put the RyR receptor in extremely close proximity to the BK channel. And this is just some data, some preliminary data we have using electron microscopy. Here again, in freshly isolated cerebral artery smooth muscle cells, where we've labeled the membranes. And as you can see in this yellow box, we zoom in, we can see that the plasma membrane and what I've highlighted in red is the sarcoplasmic reticulum. Now, this is in extremely close proximity. When we measure this, it seems to be around 18 to 19 nanometers, which is, again, trying to reduce the amount of volume that the calcium spark is released into. Therefore, it's able to generate such high concentrations of calcium without affecting everything else that's happening within the cell. And so we get this transient release of calcium, causing this calcium spark. This leads to actually a transient activation of the BK channel. And this is what was being recorded as a spontaneous transient outward current, or STOC. And so I refer to STOCs a lot of time during this talk, but just to kind of show that this was originally shown back in 1995, 30 years ago now, where Nelson and others showed that in isolated vascular smooth muscle cells from the cerebral circulation, you get this transient release of calcium that they label calcium sparks, which is closely followed by an outward current, as shown in the bottom here, which they went on to characterize was from the BK channel. And this is really quite a powerful tool for measuring the coupling between RyR receptors and BK channel, because you always get this transient activation, and it has to be driven by calcium sparks. And so we can look at a functional interaction between both RyR receptor and the BK channel in our models just by recording these spontaneous transient outward currents. So we're going to move on to the main sort of part of the talk now, and essentially that's how RyR receptors and BK channels are functionally coupled in these nanodomains. And so what I'm going to start with is kind of the structural regulation. How are these two membranes brought in close proximity to each other and interact to maintain this functional coupling? Well, we looked at essentially one of the main structural proteins, or sort of cytoskeletal proteins, and this is microtubules back in 2017. And this is in an isolated smooth muscle cell, so it's still live, it's fresh, and we use live cell dyes for the plasma membrane or the sarcoplasmic reticulum. We then imaged in to the Z stack through the whole cell, and as you can see, we can reconstruct both the sarcoplasmic reticulum and how it interacts with the plasma membrane. What we've done here is actually applied nicotinamide, which is a drug to actually disrupt the microtubule network. And what you might see from this video here, which I will play again, is that we have the plasma membrane in red and the sarcoplasmic reticulum actually retract towards the center of the cell after administration of this nicotinamide to that. And so what we think is actually that the microtubules are actually propping up or pushing the sarcoplasmic reticulum close to the plasma membrane. And therefore, when we disrupt it, we get a retraction of that towards the center of the SR, and therefore may lose that coupling. And that's exactly what we see when we've done the electrophysiology. We can see these spontaneous transient outward currents, these upward deflections as shown here. And as soon as we apply nicotinamide, we get a time-dependent decrease in the amount of activity, showing that actually we get a physical separation between the two and a functional separation. Now, we went on to show that it's not only that, we got this loss of spontaneous transient outward currents, or STOCs. But actually, when you retract the SR away from the plasma membrane, you increase the duration and spatial spread of these calcium sparks. And as you lost that BK channel feedback system, we actually increase the myogenic tone, or the contractility of the blood vessels. So highlighting the importance of this propping up the SR, and which is what it kind of looks like this, bringing it back to our original figure where we have the RyR receptors and BK channel physically coupled, where we have a microtubule network propping up that sarcoplasmic reticulum. But obviously, we're pushing the sarcoplasmic reticulum close to the plasma membrane. We need to make sure they actually don't touch. That's a really important for us because these two lipid membranes may fuse if they get too close. And so there must be a mechanism in maintaining the actual distance between the two, but also make sure there's no physical interaction between them. And so we took some inspiration from the cardiac field. Obviously, this very similar process happens in EC coupling between the L-type calcium channel and the RyR receptor, where the sarcoplasmic reticulum again has to be in extreme close proximity to the plasma membrane in order to facilitate this. A strong protein there is junctophyllin 2, which has a sarcoplasmic domain located within the SR, and it interacts with the plasma membrane and therefore maintains its physical coupling between the two membranes. What we did is we went on to show that actually junctophyllin 2 for the first time was present within vascular smooth muscle cells. And then when we've done some super resolution microscopy by looking at, in this case, at TERF, so just looking at the membrane or just below the membrane modality, we can see close association between junctophyllin 2 protein and RyR receptors, probably just below the plasma membrane. This close interaction is obviously, we think, is causing that structural interaction between the sarcoplasmic reticulum and the plasma membrane, allowing the functional coupling. So what we did is we actually used morpholinos to knock down junctophyllin 2 expression within vascular smooth muscle cells and cerebral circulation. As we can see here, using those spontaneous transient outward current stocks as a measurement of their functional coupling. In our control cells, when we depolarize the cell, we can see that we can get constant sort of a membrane potential increase in the frequency and amplitude of the BK channel. But when we exposed it to junctophyllin targeting, and again, sorry, this was after short-term knockdown, so this was 2 days in serum-free culture, we can see that we get a complete loss of this spontaneous transient outward currents, a complete loss of stock activity. What we actually went on to show is that this loss of junctophyllin had no effect on calcium sparks. But again, that was this spatial uncoupling between the RyR receptor and BK channels, very similar to what I showed previously. And again, once we lose this BK channel influence over a myogenic tone, over the membrane potential, we get a separation of the two membranes and we lose that negative feedback and increase myogenic tone. Just building it in again to our figure here, we can see that we got junctophyllin, now the squiggly green line, is actually maintaining the coupling between the plasma membrane and the sarcoplasmic reticulum. I'm not going to go into it now, but it's been shown by the early lab as well since then that actually a splice variant of STIM1 also plays a very important part in maintaining the coupling between the plasma membrane and sarcoplasmic reticulum at both these nanodomains as well as the more contractile IP3 and TRPM4 nanodomains as well. That's kind of how it's structurally put together. What we also was interested in at the time is actually how the calcium sparks occur. How are they initiated? How are they modulated? How do they change, or what processes are behind changing their frequency of firing and therefore influence BK channel activity? And so if we were to just isolate vascular smooth muscle cells and then load them with calcium indicator, so this is freshly isolated, it's not pressurized, it's not got no stimulus, no compounds in the bath, other than it's at 37 degrees. And what we see is this rhythmic firing calcium spark release. So this transient calcium release event constantly happening, and these will keep going as long as we can keep the cell alive. But essentially, what is driving this calcium release constantly is happening, a so-called spontaneous calcium release from there. And obviously, nothing is spontaneous. There's always got to be a mechanism behind driving why this is happening, what's going on. And so we thought about what mechanisms can be stimulating the RyR receptor in order to initiate these calcium sparks. So the RyR receptor I haven't got into, but it's a calcium-activated calcium channel. So it's sensitized by cytosolic increases in calcium local to the RyR receptor to cause that large calcium spark event. And so we wondered whether or not there could be some sort of mechanism driving this. If we reduce extracellular calcium acutely, we don't actually affect the calcium spark frequency or anything like that. So we don't think it's actually a extracellular event that's really driving this sort of baseline activity. We then wonder whether or not there's some crosstalk between other intracellular calcium stores within the cell, and maybe these are influencing the RyR receptors on the sarcoplasmic reticulum. What we came down to is actually lysosomes, especially, or endolysosomes. These are essentially acidic organelles that have a high concentration of calcium relative to that in the cytosol. They are packed full of ion channels, many of which can conduct calcium or move calcium from inside the lysosome to the cytosolic region. And we were really interested in this one channel here, trypamels, which I'll get onto in a sec. But in order to do this, we wanted to wonder whether or not, if these rhythmic calcium release events are there, they must be essentially stationary, or because if they were moving, like many other aspects of endolysosome and lysosomal biology, they wouldn't be in the right place to stimulate the RyR receptor constantly. And so what we can do is isolate vascular smooth muscle cells, in this here from either freshly isolated, load it with a lysosomal tracker to essentially fluoresce the lysosomes or endolysosomes that are there. And hopefully you can appreciate with the time going up that this video has not frozen. Actually, what we can see here is actually very stable endolysosomes or late endosomes within our vascular smooth muscle cells, are completely stable and therefore may facilitate the interaction between calcium release there and stimulation of the RyR receptors. We can show this is just not an artifact of our cells by just culturing the smooth muscle cells for a couple of days. And when we do exactly the same cells, we can see now we get this classical movement that everyone associates with lysosomes. As you can see here, a lot more movement in the culture cells compared to the freshly isolated. Again, showing that maybe they've lost this aspect to it. What we did show as well is actually once these lysosomes become transient and move, there is actually a lack of calcium spark activity within these cells. So we were really interested in trypamel-1 channels, a calcium release channel from the lysosomes, and maybe that could be a source of calcium that would activate our RyR receptors and initiate the calcium sparks. What we've done is we studied cells isolated from the trypamel global knockout mouse, as shown here. And when we did that, we actually saw a complete lack of calcium sparks in these cells. So there was a complete lack of interaction in both the frequency and the sites per cell. And as you would imagine, this leads to a lack of BK channel activation. We've got no calcium sparks, we can't get any BK channel activation. And again, this is recorded using spontaneous transient outward currents, where here at minus 30 millivolts, I think it was the membrane potential, we see strong outward currents in our wild type cells, but in knockouts, there is little to no activity at all. Now, in order to make sure that this is just not dead cells or there's no structural interaction between the two, what we try to do is bypass the trypamel-mediated calcium release and look for other ways of stimulating calcium influx to sensitize the RyR receptor and essentially engage calcium sparks and BK channel activity. And so what we did is we used the TRPV4 activator, GSK101. And as you can see, when we applied this to our vascular smooth muscle cells, especially in the wild type, we get an increase in stock activity and frequency. But when we've done this in our trypamel knockout mice, we actually saw a restoration of sparks or restoration of stocks. So showing that exactly that the RyR receptor and the BK channel are both functional, they're both present there, they're just not being engaged because we've lost that trypamel channel activity. And so we really kind of rescued it by trying to bypassing the trypamel mediated calcium events and then turning on the TRPV4. And this is what we had here in our trypamel knockout mice. Again, we lost the calcium sparks, we lose the stock activity, and because we've lost that hyperpolarization, we get increased myogenic tone and actually increased systolic blood pressure in these mice. And this was highlighted in an editorial based on the paper by the group in Davis. And as you can see, it kind of clearly shows that this trypamel calcium release must be interacting with the RyR receptor to initiate a calcium spark, then that hyperpolarization through the calcium elevated BK channel, driving that relaxation. And so we're kind of building in both the structural and the calcium-induced calcium release pathways into our structure just here. So we've got our tryptophan channels from endolysosomes or cytic organelles, where we get a release of calcium local to the RyR receptors in order to stimulate that calcium spark release. Again, there's some feedback that we can bypass that by activating TRPV4, which was originally shown by Scott back in 2005. But there's also other channels, and again, as shown here by the CAD 3.2 channels that was done by Osama back during his PhD, as you can show that essentially you can bring different ways of stimulating that calcium spark frequency or modulating calcium spark frequency by inducing calcium influx local to this nanodomain. What we've found as well, whilst trying to look at different ways of modulating and looking at this interaction between the RyR receptor and BK channel, we came across a MDX mouse, or a mouse of Duchenne's muscular dystrophy, where we actually found there's an increase in the calcium spark activity. Now, what we've done is isolated these cells, and what we found is not only the increase in calcium spark activity, but the size of the calcium spark was almost 50% greater. That leads to almost a doubling amount of that calcium signal that's being released. So it's released for longer, in a bigger area, and more often. And so really we're trying to understand, if calcium spark frequency is increased, is there a way of the structural regulation in actually interacting with that? What we actually found through super resolution microscopy is the RyR receptor clusters within cells isolated from the MDX mouse. These clusters were much greater in diameter and sort of size compared to that of our wild type. It wasn't just, however, just RyR receptors across the whole cell. What it was is more specifically RyR receptor clusters that co-localize with the BK channel. And so if I look down at F, which is our real main figure just here, the amount of interacting RyR receptor clusters, and what we mean by interacting, showed real close proximity to the BK channel. You can see there's a much greater increase in that area that cluster takes up, which we believe, of course, due to more RyR receptor monomers in that cluster. Now, more RyR receptor monomers in the same cluster will lead to an increase in calcium-induced calcium release activity, because you've got more monomers there sensing a local environment, but also translating small calcium release events into a bigger one by sensitizing its local partners within that cluster. So this MDX mouse not only had, again, this increased RyR receptor activity, we showed that an increased spatial frequency of these calcium sparks, but increased spatial spread. There was no effect on the coupling between the two. What we showed was a massive increase in their stop frequency because of the increase in calcium sparks. This decreased the myogenic tone, so it boosts the amount of BK channel activity and therefore dilated or caused a larger blood vessel within there. And this was highlighted also in an editorial by Christian Soler in Bern. And this is where, again, showing that increase in cluster size drives increasing the amount of calcium spark activity and therefore hyperpolarizing the plasma membrane via BK channels. So that was a lot of the research that I've done, especially during my time in Nevada, but also bridging that into Manchester, where I moved back. But again, as I mentioned at the start, I'm really interested now in trying to look at more of a disease angle to that. As most people be well aware that dementia is obviously almost reaching epidemic levels now, and there is no treatment for that. And so recently we've been turning our attention to dementia models and can we study ion channel activity within there. As I said before, dementia cases in the world on the rise worldwide, and actually it's the leading cause of death now in the UK. And collectively, vascular dementia and Alzheimer's disease are a main player of this. And again, what I wanted to talk about today was how hypertension and sort of vascular dementia can occur within majority of patients with this. We're really interested in dementia because there are essentially 40 modifiable risk factors for dementia, which, again, being a vascular lab, we would argue at least 8 or 9 of these are driven by vascular dysfunction. This is obvious in, say, traumatic brain injury or sort of high cholesterol. But what we're really interested in at the moment, or the last few years, has been hypertension and how this impacts the blood vessels within the brain and how that progresses onto sort of disrupting the blood flow to the neuronal tissue and therefore cognitive impairment. And so really, in order to study dementia and essentially hypertension and how it leads to dementia, we had to use a mouse model. The mouse model we chose was the polygenic BPH-BPM mouse model. We're really interested in this mouse model because, again, it's polygenic model of hypertension, hypertensive from a young age and throughout life, but it has only a modest increase in blood pressure, around 25 millimeters of mercury, compared to other models that are quite extreme in their process. They've been shown before as well to have impaired neurovascular coupling, and in our publication, I'm not going to go through the data now, but we showed that these mice are cognitively impaired and they have a reduced amount of blood flow, just basal blood flow to the brain, signifying that there is some sort of vascular dementia phenotype happening. When we take these arteries from the hypertensive mice, what we found is actually their autoregulated properties are actually overconstricted. This is as shown here by where we can essentially take the blood vessels out and increase the intranominal pressure by turning up using pressure myography, and we can look at the response of these blood vessels to that pressure. If I just talk you through the normotensive one here, so as we increase intranominal pressure from 10 up to 80 in a stepwise manner, we can see we get an active constriction of the blood vessel over at each intranominal pressure, which essentially we're trying to mimic autoregulation in the lab. What we found is that the blood vessels from the hypertensive mice have a more exaggerated constriction over that blood pressure range, and as you can see here, what we think might be happening is a lack of autoregulation and therefore feedback potentially from the BK channel. We think that as well is because when we maintain sort of a flat diameter or a single pressure, in this case it was 60 millimeters of mercury, we get generation of myogenic tone. We can then apply an inhibitor to the BK channel, in this case paxillin, and we get a vasoconstriction in a normotensive mouse as shown here. So this is losing that negative feedback system in the presence of the drug. And we can determine that amount of constriction is actually the amount of BK channel influence over the membrane potential and therefore diameter of the artery under myogenic tone. When we did this in our hypertensive, we see that it's almost half the amount of response to paxillin, suggesting that potentially we've lost some BK channel influence over that in our hypertensive mice. Again, we go back and we do our electrophysiology. Again, freshly isolated vascular smooth muscle cells from the pure circulation, recording spontaneous transient outward currents. What we found is actually there was no difference in the amplitude that these are generated in these hypertensive animals. What we found is there was a marked decrease in the frequency of firing between our hypertensive cells from our hypertensive mice compared to that of the normotensive. And so we kind of went through potential mechanisms that might be behind this impaired BK channel dysfunction in hypertension. First one we thought about is potentially, okay, if we're losing calcium sparks, this may be due to a loss of renin receptor expression, impaired calcium-induced calcium release events, or even store low the amount of calcium within the sarcoplasmic reticulum. We would lose calcium sparks, and therefore we would lose interaction between, and therefore the activation of the BK channel. We may actually lose BK channel itself. And so if we lose the BK channel or even this calcium sensitive subunit, the beta-1 subunit, we may no longer get that. So we might be getting calcium sparks, but not actually getting channel activation because they're not present. Or a third option may be that we're actually getting a breakdown in this peripheral coupling site integrity, the interaction between the renin receptor and BK channel, maybe functionally and physically uncoupled. In the interest of time, I'm not going to say that the calcium sparks were completely normal between the two models. So our normotensive and hypertensive animals showed exactly the same amount of calcium spark frequency and dynamics and kinetics. When we looked at wholesale BK channels, so we bypass renin receptors and directly activate the BK channel itself. Here we're looking at paxillin sensitive currents in a clamped calcium concentration here at 500 nanomolar, looking at minus 100 to plus 100 millivolts. What we actually found, kind of serendipitously, is actually there was an increase in current density in cells isolated from a hypertensive mice compared to that in a normotensive. Essentially, the BK channel is there, it's functional, it's just not being functionally interacted with by calcium sparks. One option could be, again, as I mentioned before, that maybe we've lost that calcium sensitivity. So again, we can use those freshly isolated cells. In this case, we're doing inside-out patch and look at various concentrations of cytosolic calcium to that and see what happens to the open probability as we step it through. And again, I just wanted to highlight that again, we need around 10 micromolar calcium before we start seeing any channel activity, especially in our normotensive cells from our normotensive mice. But when comparing between the groups, there was no difference at any calcium concentration, suggesting that we probably have beta-1 subunits functionally interacting with our alpha subunit, the BK channel, but they're just not being activated by these calcium spark events. So that brought us to whether it was a breakdown in the peripheral coupling site integrity, and it's more of a structural story. Again, we went back to our stuff that we'd done back in 2017 in the early lab, where we can isolate cells, live cells, and use the membrane dyes for the plasma membrane or sarcoplasmic reticulum, and compare that to between the normotensive and hypertensive. We can then merge them and look at sites of co-localization to see whether or not that happens. We didn't just do it in one slice, we done it through a whole 3D volume. And using Amara software, we can rebuild this and try and essentially build that plasma membrane area, sarcoplasmic reticulum area. But again, those two sites of co-localization, we're calling them peripheral coupling sites. We created a third channel to that. And this is where we can look at the interaction between the two across the plasma membrane, and we can record the amount of interaction, size, volume, etc. When we compared this between our normotensive and hypertensive mice, there was no change in plasma membrane volume, SR volume. What we found was the number of peripheral coupling sites per cell, and also the volume of each peripheral coupling site was markedly decreased in cells isolated from our hypertensive animals compared to that of the normotensive. Again, this doesn't quite say, this is just the two membranes. It doesn't say whether the two proteins are physically uncoupled. And to do that, we use proximity ligation assay. This is where if two proteins are located within 40 nanometers of each other, you can create a chain reaction, essentially find a purple spot within our cells. And this is what we do in our normotensive cells. So here we have positive proximity ligation assay from a 3D reconstruction of the whole cell, counterstain for the nuclei. And as you can see, there's multiple sites per cell where we get physical interaction between the RyR receptor and BK channel, and therefore our calcium sparks and stop activation. When we've done this between our hypertensive and compared that to our normotensive mice, what we found is that there's actually a massive decrease in the number of puncta per cell in cells isolated from our hypertensive animals compared to that of the normotensive animals, which suggests that there's physical uncoupling between the two membranes and functional uncoupling. And so that was our hypertensive story, where we found that actually this loss of peripheral coupling site integrity really drives that BK channel dysfunction. Our hypertensive mice displayed an impaired cognition. I haven't shown that today, but again, just reminding you that there's a reduction in cognition and cerebral blood flow. And this is due to a hyperconstrictor blood vessels, due to an inability to autoregulate the arterial diameter. And this is due to a lack of BK channel activation, leading loss of this peripheral coupling site and that interaction between calcium sparks and BK channels. And that just brings us back around to the take-home messages to kind of repeat what I've just saying, is that constant brain blood flow is maintained by autoregulation. BK channel is vitally important in maintaining that larger diameter and therefore correct flow. There's a complex regulation and structure between the two that influence this, and it is damaged in hypertension. And just bringing it all the way back to our structural and calcium-induced calcium release mechanisms, this real complex mechanism maintaining the important coupling between the RyR receptors and BK channels is shown here. With that, I'd like to thank everyone at Manchester with the group that we're developing here, but also Scott Early in the previous lab, who's now moved to the University of Rochester, and the previous members of his group there. And again, I'd like to thank the members of the current lab and Scott for all their support over the years. And with that, I'm happy to take any questions. Thank you very much.
Thanks, Harry. Great talk. Really wonderful. Taking us through kind of the different studies leading to this. We already have a few questions in the Q&A, so I will read to you from these. So the first question from Rohit Kumar, can we really use the Poiseuille law here, because blood flow in vessels doesn't necessarily follow the conventional idea of pressure difference across two ends of a tube?
Yeah, I can understand that kind of ideas. I think you can to a certain extent because there is a pressure difference between the two. There's pressure difference between that coming out of the heart and obviously the resistance that's down in the blood vessels in the organ, in this case, the brain. And so there is a pressure difference between the two, and there is a flow system. It may not be perfect. We don't have a proper way of doing that, but I think it's a good approximation of what's actually going on here. It's obviously as well, it's quite clear that if we change the diameter of blood vessels, we can increase blood flow further down. And we can do that in our in vivo models. I'm sure I didn't show it today, but we can dilate blood vessels and show that actually you get, by dilating the blood vessels with, say, hypercapnia, you can essentially drive the amount of blood flow to the brain and increase that flow downstream. So changing diameter does change flow, and therefore that's the approximation that we're getting into it.
Thank you. The second question from Paul Wagner. There is a known functional coupling of calcium sparks and RyR cluster size. Did you see a co-clustering of BK channels as a mirror image of the RyR, or do they behave differently?
Actually, I think they behave differently, especially we take that from our MDX study where we showed that we get an increased amount of BK channel size, although we get increased cluster density, especially in our RyR receptors in the sarcoplasmic reticulum. There was no change in the BK channel cluster size of the plasma membrane. So although there is clusters between the two, where we get bigger clusters of RyR receptors, we get bigger clusters of BK channels. There's not like a one-to-one relationship between the two, and so they're not recruiting more BK channels, even though there's a bigger RyR receptor cluster size there.
Yeah, another question from Rohit Kumar. Can we really consider hypertension-based loss of memory as dementia? And how would that fit with pathophysiological side of a cell that has a molecular background across the line as subject ages?
I would argue that so we're very interested in what drives dementia, especially current research. What we found, especially from our working with clinical colleagues at Manchester, is that hypertension is one of the main, essentially midlife hypertension is one of the main risk factors driving cognitive decline as in later life. So hypertension may not be the sole driver of dementia. We obviously know that amyloid and Alzheimer's disease is a big component of that. However, we're always saying is that actually there's a hypertension will disrupt vascular physiology, and therefore if you reduce blood flow to the brain, as we know from other things like stroke and everything else, that we will impair neuronal function. I don't think it's going to be the sole driver of dementia, but I do think it's a good component of that.
Question from anonymous attendee: Are SK and IK channels influenced similarly, similar to the BK as you saw for BK channels?
Yeah, so this is something I've always thought of as well. There's no IK-SK channels in vascular smooth muscle cells, but they are closely located in the endothelium where they're activated by IP3 mediated calcium release more. As far as I know, no one's really looking at that kind of structural regulation, what's happening, how close they need to be in order to activate the channel. Something I thought about, I should kind of move into that as well. Just kind of trying to balance the amount of impact and obviously what I can do with two sets of hands. And so we're trying to work out what we can do. But yeah, I'm very interested in that. We don't know entirely, to be honest.
A question from Damien Bell. TRPM-L1's role in vascular dementia, including Alzheimer's and other neurological diseases like Parkinson's, is often suggested to be due to lysosomal clearance of neuronal proteins disease. Your lab investigated BK coupling. Could another be reduced TRPM-L1 activity?
Yeah, exactly. Yes, I do think so. I've always thought about the role of lysosomal storage diseases, big thing, especially in the dementia world. And so the interaction at the neuronal level, obviously, the storage diseases, that dysfunction, a component of that as well, especially if we're getting lysosomal storage disease affecting the vascular contractility. If we're moving TRPM-L away from these RyR receptor nanodomains, we could be impairing blood flow, and therefore affecting not only the neuronal function, but the supply of energy because we're reducing the amount of blood flow to that region. So yes, it's something I've always thought about. It could be something happening there. Again, not quite got around to doing that one yet.
Another question, which brings a smile. Anonymous attendee, is there any role for the endothelium in these interactions?
Endothelium is a definite feedback mechanism into it. Obviously, it senses a lot of different inputs from various different aspects of vascular physiology. Hyperpolarization, depolarization can be transmitted to the vascular smooth muscle cells and then influence contractility. Whether they directly interact with the coupling between the BK channel and RyR receptors, I don't think so. But again, that conducting that hyperpolarization across into the cell would definitely influence contractility and therefore potentially lead to different changes in diameter.
Question from Asser there. Great talk. What could underlie the structural uncoupling in hypertension? In other words, can it be targeted therapeutically?
Maybe you got my last grant. Yes, that's exactly what we're trying to do at the moment. So we've worked out, essentially, we're losing junctophyllin in these peripheral coupling sites. We are trying now to restore the integrity of junctophyllin, see if we can maintain that and whether that improves blood flow to the brain using a pharmacology approach. As it was being recorded, I didn't want to show any preliminary data yet, but more than happy to share offline if need be.
Sounds great. Question from John Lederer. Beta amyloid question. Any role for beta amyloid? How do you explain counter examples? I'm not sure.
Not sure. Sorry, would you say that again?
So the first question is about beta amyloid, whether beta amyloid has any role, I think, in the coupling aspect.
In the coupling aspect, yes. So we published a paper 2 years ago showing that, especially in the AD mouse, APP23, that we lose calcium sparks. We're trying to work out how that happens, but essentially we think it's a lack of calcium within the cytoplasmic reticulum, so a decrease in calcium store load. Beta amyloid itself can actually activate calcium events within the sarcoplasmic reticulum of vascular smooth muscle cells, and what we've shown actually is induces calcium waves. So these are propagating calcium signals that run up and down the cell as soon as it's exposed to nanomolar concentrations of amyloid. It may be stealing essentially calcium away from the RyR receptor sort of pools, and therefore may decrease BK channel activity. But as we've been shown before, if we put on amyloid beta, we can constrict blood vessels. It kind of generates more of a calcium wave more than a calcium spark. And so there is definite interplay between the two, and we're really interested in how that happens.
Sure. John just followed up on the question because I didn't understand it. So he's saying that there are people with excess beta amyloid genetically without disease, as this is the counter example. So yeah. Really, whether that's really going to be the game-changing kind of component of it. So.
Okay, sure. So yeah, amyloid is a strange one. We're still working out exactly what happens. I think it's a lot of where it is and what's it interacting with is the main thing. What we've seen a lot recently is that people may have high amyloid load, but if they can clear it efficiently, then it won't interact with the neuronal space or even the blood vessels themselves. And so there is some impact of how we detect amyloid. Is it in the CSF, stuff like that? Actually, you can have a high amyloid load and be cognitively sound, mainly because you're clearing it into the CSF and therefore it's detected. If it's left behind in the blood vessels in the neuronal space, you might actually on a test see a low amyloid load and therefore maybe confused between the two. Again, it's a great question. We're still working on what's happening between both. We don't know. We don't have the answers. If we do, then we'd be a lot further ahead, especially in disease treatment. But it's a very important point, and thank you for that, John.
Frog Wagner, thank you for the great talk. Are there any known pathologies affecting junctophyllin 2 that leads to hypertension?
Yes, great question. So I've looked at this, and there are mutations associated with junctifilin 2. Unfortunately, none of them have really been looked at in the hypertension phenotype. As I mentioned in my talk, junctifilin plays a massive role in EC coupling and maintaining cardiac physiology. And so normally if you get a mutation or a dysfunction in junctifilin 2, I don't think you will live long enough in order to get that kind of hypertensive phenotype with age, mainly due to cardiac defects that happen. And so we are looking at different ways of trying to look at if there's any sort of modest slight mutations in junctifilin 2 that might lead to impairment of vascular function away from the heart. But I'm afraid nothing's been reported yet.
Skuzavia is saying, thanks, Harry, great work. What drives BK recruitment or retains BK to the membrane contact sites?
Again, great question. So we know that BK channel normally resides in kind of lipid rafts and caveoli. And so we think it's actually been recruited there, potentially by cholesterol or other proteins around that caveolin 1. But actually how it's maintained in the membrane is something that I'm not too familiar with, to be entirely honest, but we know that the interaction is vitally important with caveolin 1.
That's great. Another question from Boris Lavenders. I'm just curious about how hypertension causes the uncoupling and whether you have looked into other causes of dementia.
Yeah, so as I mentioned earlier, we're kind of looking at different of how it's caused this uncoupling. We're very keen on essentially junctifilin is being essentially chopped up and removed from these coupling sites. And so we're looking into how that happens and can we restore that function. But again, I'm not just a BK channel person. We look at any ways of maintaining arterial diameter control at the right down deep at the capillary network, but as well as up at the pure circulation. And so essentially we are looking at different mechanisms that could be happening that may also be targetable as well. I don't think it's just a BK channel story or just a junctifilin story. There is a whole range of pathologies probably going on and damaging blood vessel function that may be targetable.
A question from Wasim El-Khatib. Thank you for the great talk. Does calpain regulate smooth muscle function in the same way it does in cardiomyocytes?
Yeah, so that's exactly what we're doing. So calpain will chop up junctifilin 2. And so that's currently what we're looking at now, is that calpain regulation of junctifilin 2 and how that interacts with dysfunctional BK channel dysfunction, in hypertension.
Sure. A question from John Willis. Are hypertensive mice mentally deficient or cognitive?
So yeah, they are cognitively impaired. I didn't kind of spend too much time on that today, mainly because of what is more of an ion channel overview. But these mice are cognitively impaired. They have novel object recognition tests. They were impaired at that. They was impaired in the kind of open field maze aspects. They essentially are cognitively impaired as much as we can take from a mouse model. But yeah, they show some symptoms of that.
Sure. John Litter again says, great talk. Manuel Nevedo is saying, great talk, Harry. Are the changes in BK-RYR similar in female cells? Is there a sexual dimorphism kind of phenomenon here?
Yeah, so we've also done this in the female BPH mice now. They are impaired. It's actually, I think both are impaired. And this should be a greater effect in female mice than there are in male mice. What we've also found is actually in our normotensive mice, there's also impairment as well. So we're looking at, as the mice age, in both male and females, whether or not treatment will actually restore this in both sexes as well. There is some counterbalance to that. I know other studies, even in our aged AD mice, we actually, in our normotensive controls, we see an increase in calcium spikes as they age. So a bit wary about what's happening in our BPM mice going forward. If anything, we are actually slightly moving away from the BPM mice. But overall, yes, BK channel dysfunction is driven as well in the female mice.
I think the last question from an anonymous attendee. Great talk. Did you observe a similar dominant role of BK channels in the myogenic tone regulation of PAs penetrating arterioles? Or did you examine only the PL vessels?
We only done the pills so far, but yeah, other groups have looked at BK channel function in parenchymal arterioles. They seem to have less of a regulatory role. Work from Fabrice de Bertrand has shown that actually there's very little BK channel influence over parenchymal arterial tone. But we haven't looked at that in our hypertensive model yet.
Cool. Last comment from Scott early. Great talk, Harry. So, and I echo this.
A bit biased though there.
Yeah, no, really great talk. Thank you very much again. We are a couple minutes beyond the hour, but thank you again for accepting the invite and for a great talk and really very engaged question answer session. So thank you again for a great talk.
Thanks everyone. Thanks for the feedback as well. It's really great.